So this video is going to be a basic introduction to cell division. Where we're going to start this unit is with vocabulary. There is a lot of terminology in this unit, so we want to kind of lay the groundwork for what we're going to see in terms of language when we talk about cell division. So here's a brief overview of some of the terms you're going to see in this unit. So we'll talk about all of these in context. This is not the only time you're going to see these words. You'll see them lots as we go through and start looking at the processes of the cell cycle and cell division. In addition, some of these should be reviewed. So nucleotide, we've already seen before. Chromosome, things like this might be familiar from middle school. So if you took a look, kind of when we went through briefly, at all of the words we had for this unit, a lot of them revolve around DNA, which begs the question, why? So why is it that we spend so much time at the beginning front-loading all of this vocabulary around DNA? And it has to do with the fact that all cell division is a form of reproduction. When a cell divides, it's making more of itself. Now, the reasons behind why it's doing this or the intended purpose is going to vary depending on whether we're looking at mitosis, meiosis, or even later on with prokaryotes, binary fission. But in each case, we're making more cells. So the cell cycle, we talk a lot about DNA because it's preparing the cell to divide. In mitosis, this is about eukaryotic cells that are asexually reproducing. And meiosis is about creating those sperm and egg cells for sexual reproduction to occur. Regardless of which type of division we're looking at, though, we know that DNA is going to contain the information necessary to functionally run a cell and create more of the species. So in mitosis, we want to take a cell that's totally functional somewhere in your body. It's doing its job perfectly. So for example, a liver cell. So right now there's a cell in your liver. It's a rock star doing exactly what it's supposed to do. But that cell's not going to live forever, which means that it needs to replace itself and divide to create two new daughter cells or offspring that will be able to pick up where it left off, continue being rock and liver cells. In order to do that, it's got to pass on all of its genetic information, all of its info to how to be a liver cell. And in order to do that, we got to pay attention to what's happening with the DNA. Reproduction also relies on DNA. So why is it that our chimpanzees always have chimpanzee babies? Because they're passing on their genes to the next generation. So this is what meiosis is going to show us. How is it that humans always have human babies? Why is it that chimps always have chimp babies? So we're going to look at how the DNA is moving from one generation to the next. We're going to start our investigation into cell division by looking at the organization of DNA inside of organisms. We'll start with the largest level of organization within an organism, and that's called the genome. So all of the genetic information in a single organism is called the genome. So for example, back in the early 2000s, there was something called the Human Genome Project, where a group of scientists from around the country were looking at sequencing all of the A's, T's, G's, and C's in a single human being. So they wanted to know, what is the whole code to make one person? Now, the interesting thing about the genome is that for most organisms, and humans are definitely no exception to this, a lot of your DNA is not actually used. It's actually referred to as junk DNA. So not everything that's in your genetic code is actually going to be used by your cells. A lot of it's leftover garbage. So some of it could have come from a virus that left some DNA behind when it inserted itself into a cell. Some of it could be genes that maybe were used a long time ago by our ancestors, but now have been turned off and don't get used anymore. So this contributes to this kind of junk. The next level that we have are our chromosomes. So within our whole genome, we have these condensed structures. Now, the interesting thing about eukaryotes like humans is that our DNA is linear. So it comes in these straight lines. And in humans, we have 46 straight lines of DNA, so 46 chromosomes. Within those chromosomes, you have these sections of DNA that code for a protein. And we call this section that contains a recipe, these are your genes. So essentially in genetics, what we're studying is we're studying the recipes in an organism. So when we study your eye color, for example, we'll look at your 2 copies of that chromosome that contain eye color. We'll see what recipe is on one chromosome, what recipe is on the other, and then can we make predictions about what your eye color is going to be based on those genes. And again, remember, there's junk DNA, so not all of your DNA is actually coding for proteins. Quite a bit of it's not. And then the smallest level, so looking at what makes up our genes, are nucleotides. So nucleotides are those monomers or they're building blocks of DNA or RNA. We did this in organic molecules. They're made up of one phosphate one sugar. In the case of DNA, it's a deoxyribose sugar. In the case of RNA, it's a ribose sugar, and then a nitrogenous base. Because the phosphate and the sugar are a constant, we don't actually use them when we talk about the genetic code. Remember that the variation in the code, so what stores the information, those are your nitrogenous bases, which is why when we look at the genetic code, it's literally just the A's, T's, G's, and C's, so the adenines, thymines, guanines, and cytosines. We don't actually talk about the phosphates or the sugars because they don't change. The common analogy that's used to understand the organization of DNA is a book analogy. So you can think of the entire information to make an organism is like a book. And that book has the whole story. It's got everything you need to know. But books are broken up into chapters, and chapters help us to identify certain sections. So it's the same thing with our genome. So we can point to the addresses of certain genes and certain information by looking at what chapter it's in or what chromosome it's on. Within chapters, we have genes. Just like within chromosomes, we have these sections that code for proteins. And then we know that paragraphs are made up of letters. And the letters or code of our DNA is going to be our nucleotides. So again, those A's, T's, G's, and C's. So that's an analogy for helping to understand the organization of DNA. Now we're going to use that information about the structure of DNA to talk a little bit about the DNA of our cells. But in order to bring these two together, we first need to talk about the two types of cells that we'll be discussing in this unit. Now, these two different types of cells exist only in sexually reproducing eukaryotic cells. Now, I know that's a mouthful, but you'll understand kind of why it has to be sexually reproducing in a second. So the first type of cell that we find in these organisms are called somatic cells or body cells. So the term somatic you will see a lot, and again, this just refers to any cell in your body. So the example I have over here, a neuron is in your body. It is a somatic cell. A red blood cell, a muscle cell, a white blood cell, the columnar epithelial cell, so your skin cells. These are all in your body. So the other type of cell we have is much easier to remember kind of what is a sex cell, and then everything else that is not a sex cell is a somatic cell. Sex cells, there's literally one in a biological male, and that is the sperm, and there is exactly one in the biological female, and that is an egg. Every other cell in a biological male's body that is not sperm is a somatic cell. Every other cell in a biological female's that is not egg is a somatic cell. Doesn't matter where it's in. It can be deep inside your liver. It can be on the edge of your skin. It is a somatic cell if it's not sperm or egg. We have a name for these sex cells. They're also called gametes. So gametes is a generic term to refer to these sperm or egg or these reproductive cells that we have inside sexually reproducing eukaryotic organisms. When we look at the chromosomes, so again, those chapters, those organization of genetic information in somatic cells, so in your body cells. So if we looked in your liver in a nerve cell, we would find that you have 23 pairs of chromosomes, giving you a total of 46. This diagram over here on your right-hand side is referred to as a karyotype. We will look at these quite a bit when we do genetics to look at diagnosing genetic conditions. And that's what these are used for. So to see if there are any errors in the chromosomes of a developing human. So here we can see those 23 pairs of chromosomes. So just a quick question, see if we can kind of figure this one out. Where do you think these 23 pairs came from? So why are they pairs? Hopefully you were able to get this. So they're pairs, which means they're coming in twos. We're looking at sexually reproducing organisms. So what you're essentially seeing here is you're seeing for every single chromosome, you got one copy from dad inside the sperm and exactly one copy from mom inside the egg. So people tend to have this thing where they're like, I look more like my dad, I look more like my mom. Now that might be your gene expression, but you are literally exactly 50% DNA from mom, exactly 50% DNA from dad, because each parent contributes exactly one copy of every single chromosome inside their gamete. And these combine to give you what you have in your somatic or your body cells, those 23 pairs.